All right, hello biochemistry class. We are here to start talking about chapter 5, which is going to be all about the primary structure of proteins. So the primary structure of proteins refers to the order of amino acids in that peptide chain. But before we can even start talking about primary structure, we want to talk more about how we can even get a look at the proteins. And the way we're going to do that is looking at protein purification techniques as well as protein characterization techniques that help us to extract out and purify individual proteins and then characterize these proteins by their properties. So first, in this section of the lecture, we're going to talk about protein purification. So as an overview for protein purification, first we of course need a source of our protein. So if you're looking for a protein that naturally occurs in an organism, that's one way you can start by using either a tissue from an organism or individual cells. For example, if you're looking for a protein that naturally occurs in a bacteria, you could isolate that protein from that bacteria. Or if you're looking for a mammalian protein, you could get mammalian tissue and isolate out that protein. But the one issue with using natural sources of proteins is that there are so many other proteins that are occurring in the cells and tissues of any organism. So your protein of interest, your POI, is usually can be even under 0.1% of the whole tissue or cell that you're lysing. So using a natural source, you can potentially end up with very low yields of your protein. And so you'd have to start out with a massive quantity of tissue or cell material in order to get a good yield at the end of your purification process. Another method that's often used in the lab is using a genetically engineered source of protein. So for example, you can use a specialized expression strain of E. coli to specifically grow your protein of interest. And that way, when you then lyse open those cells to purify your protein, you can already be starting out with up to 40% of your protein of interest as the whole component of cellular material that you're starting with. So even though it's a lot more work to set up a genetically engineered source of protein, the reward can be very great. And so it's a fairly common lab technique for people who are trying to work with and purify proteins. So either way, whether you start with a natural source or a genetically engineered source, you often in the lab need to carry out multiple purification steps to remove other proteins that are going to be in your sample. So even with a genetically engineered source of protein, of the cellular material, maybe you have 40% of your protein of interest, but you still might have 60% of other proteins that you don't want there. So there are many purification steps that you can do to purify out just the protein that you want to look at. And over the course of all of these purification steps, it's really important to protect the protein of interest from damage. So while you're in the lab, you need to do things like control the pH and the temperature, and that because varying pHs can make proteins more or less soluble based on their pKa's and pI's. At higher temperatures, proteins are more likely to degrade as compared to lower temperatures, and that's why we store a lot of our samples in the refrigerator or the freezer. Also, whether your protein comes from a natural source or a genetically engineered source, there are going to be degradative enzymes that occur in that cell or tissue source. So you need to protect your protein from those enzymes that might break down your protein. And even a lot of proteins are sensitive to air and oxygen, so you need to always keep your containers covered. So that's just a number of different things that you need to look out for over this process of purifying your protein of interest. And finally, after you're done with the whole purification process, then you'd be ready to finally assess the success of your purification by doing a number of different types of characterization steps that we'll go into in other lectures. So let's use as our example this PUG18 plasmid that we talked about a couple of lectures ago. So this is a plasmid that we might insert into an E. coli, and we'll use this plasmid to instruct the E. coli to make our protein of interest. So there are a couple of different parts on this plasmid that we've discussed before. So the first one, we have our AMP resistance gene. This is our antibiotic resistance to ampicillin. And so if we are able to grow our E. coli in media that contains ampicillin antibiotic, only the E. coli that have this plasmid inserted will be able to survive and propagate. So that helps you select for only the cells that are going to help you make your protein. And the other area of interest is this LAC-Z gene that contains the polylinker region. So the polylinker region is where you will insert your protein of interest. And then the LAC-Z gene, and in fact the whole LAC operon, is really important for instructing the E. coli to start expressing your protein. So let's talk a little bit more about that. So the LAC operon consists of a number of different parts. So this sort of rectangle area at the top sort of represents the DNA code, the genetic code for the various genes in your LAC operon. So the first gene on the left in pink, we have the LAC repressor. So that's the LAC-I gene, and it codes for the LAC repressor. And so when the LAC-I gene is expressed, the LAC repressor protein will bind to another region on the LAC operon and actually stop the rest of the LAC operon from being transcribed and therefore translated into protein. And so it's called a repressor because it stops the other genes from being expressed. After that, we have the promoter, the operator, and then the LAC-Z gene. The LAC-Z, as we also discussed a couple of lectures ago, codes for beta-galactosidase. This is the enzyme that will break down lactose. So the LAC operon, its main job is to be expressed when the bacteria need it, when there's lactose in their environment, and they want to be able to digest and break down that lactose. So that's why the LAC operon codes for this gene that helps to break down lactose. And that's why we have this repressor. The repressor is there because most of the time we want this gene turned off. We only want our beta-galactosidase genes and the other genes that are in the LAC operon to be turned on in the presence of lactose. Otherwise, it's essentially kind of a waste of energy and time to express all these genes, just to transcribe them into RNA and to translate them into protein. You're sort of wasting materials and energy. So you only really need the LAC-Z, LAC-Y, and LAC-A genes in certain environmental conditions, and that's why we have this whole operon system. So that's a bit of a digression, but anyway, that's the LAC-Z gene. It codes for the beta-galactosidase enzyme. And then there's also the LAC-Y and LAC-A genes that are part of the LAC operon. They're more important for the actual function of digesting lactose for the bacteria. They're less important for our actual purposes in the genetic engineering of E. coli. So for our purposes, when we are working with the LAC operon to help express our gene of interest, we really work with just the beginning part of the LAC operon. So the LAC-I, which is coding for our repressor, and the LAC-Z, which is coding for our beta-galactosidase enzyme, as well as the promoter and operator in the middle. So under normal conditions, what would happen is the RNA polymerase would come along and bind at the promoter region, and then it would start to transcribe RNA, going 5' to 3'. It would pass over the operator and the LAC-Z gene, and ultimately end up transcribing the LAC-Z gene into LAC-Z RNA, which then gets translated into beta-galactosidase. So that's great. And but the sort of functional part of the LAC operon is, of course, our LAC-I gene, which codes for the LAC repressor. So the LAC repressor, that part is always sort of on and always being expressed. So when we actually have these LAC repressor proteins, they bind at the operator region, and they actually physically block the RNA polymerase from passing over that operator region over the code for the LAC-Z gene. And so once the LAC repressor gene is down there, the RNA polymerase is no longer able to transcribe our LAC-Z gene. And so we no longer are able to make our beta-galactosidase protein. So let's reset that. So what happens when there's actually, lactose in the environment and we need to turn on the LAC-Z gene and we need to break down lactose? Well, the normal sort of way that would happen in the environment is when there's the presence of allolactose. Allolactose is a form of lactose that actually binds to the lac repressor, and it actually keeps the lac repressor from sticking to the operator region. And so if the lac repressor is no longer blocking the operator region, now the RNA polymerase is free to go on and transcribe the LAC-Z gene, which makes it which makes then RNA available to get translated into beta-galactosidase. And now beta-galactosidase is there and is able to break down the lactose in the environment. Bacteria can use it to, make more energy for itself and go on and live its life. So let's go ahead and reset that. So how do we use this in the lab? So in the lab, instead of having our LAC-Z gene over here in green, we have our gene of interest. So under normal conditions, when we're just growing up, our E. coli, our lac repressor gene is being made. It's blocking the operator region. RNA polymerase is not able to make our gene of interest. But when our E. coli is in the correct growth phase and we're ready to start asking it to translate our gene of interest, we add an inducer. And the inducer that we use in the lab is called IPTG. And IPTG is an analog of lactose that is really much more stable. It's not cleavable, so it's always around and it's always there to block the lac repressor from binding to the operator. And if the lac repressor is unable to bind to the operator, now, once again, our RNA polymerase is free. It's free to transcribe, and now it's going to transcribe for us our gene of interest. And then our gene of interest, RNA, is going to be able to be translated to actually make the protein of interest that we want to purify. So this whole process of genetic engineering and using the lac operon is what helps to ask our E. coli to make for us lots and lots of copies of our protein of interest and massively overexpress it. And that's due to our inducer IPTG and this whole lac operon system. So once we've actually made our protein of interest, we have maybe all these E. coli cells. They're full of our protein. How do we get to our protein? So the first step of that is going to be homogenization and cell lysis. So in the case of bacteria, they're already single individual cells, so we need to work on just lysing those cells. In other cases, you might be working from whole tissue, from maybe a mammalian source or other source, plant source. So for those first, you generally want to homogenize your tissue and then go on to further lyse those cells. So there are a number of different options for all of these processes. So the first way is pretty straightforward. You've probably seen one of these before, is a blender. A blender is a really good way to start breaking down tissue for you. The next option we have, and that's a blender. Hopefully you know that. The next option we have is a sonicator. So the sonicators often look a little bit like this. This is a probe sonicator where you stick a probe into your sample, and it uses essentially sound waves to help break open cells. So you'll often use this when you have a culture of bacteria or something that's already a single cell suspension and you just want to break those cells open using sound waves. After that, we have the freeze-thaw method. So freezing and thawing is a very straightforward way to break open cells. You go through multiple cycles of freezing them and letting them thaw, and the formation of ice crystals and going back and forth between ice and liquid water essentially just bursts cell membranes and cell walls open. The next option is a French press, which is drawn over here. And that essentially just uses pressure to force all the cells through a very tiny hole that helps to sort of squeeze the contents out and break the cells open. And then one last option we have here on the slide is the mortar and pestle, which you may have seen, in the kitchen or somewhere else. But we also do use it in the lab to just physically help grind up samples and start breaking them down. And these are just a few of the options that we have here for homogenizing and lysing the cells. So once you've broken the cells open, now we want to start thinking about how to really pick our protein out from all of the other, unwanted stuff that's in the cell, like the cell walls and the other organelles, and of course that would be for eukaryotic cells, or just other cellular components that you don't want. So a basic first step is using centrifugation. So this is really describing, I'm now going to go forward describing a process for purifying out a protein of interest that is soluble. So if your protein of interest is soluble, if you centrifuge your lysed open cells, right, you put your sample in a tube, you put the tube in a rotor. The rotor then spins very fast and it separates out solids from liquids. So solids are denser, they'll end up in the bottom in a pellet. And this will contain most of the insoluble components of the cell, such as cell membranes, other sort of lipid components. The supernatant is going to contain all the soluble proteins, including our protein of interest, and also other components that are soluble. So that's a general first step, and so from there, as long as you would pick out the supernatant from there, save the supernatant, discard the pellet, and now we have this liquid portion that contains all of the soluble components of our lysed cells. So the next fairly common method to then start separating out your protein of interest is to use solubility. And the method that we often use for this is called salting out. So in this picture here, we're depicting essentially the supernatant after our centrifugation step. Our supernatant is full of a bunch of different proteins that are soluble. Our target protein here is green. There's also, in this picture, red ones and yellow ones, and we don't really want those, right? We're trying to purify out just our protein of interest. So what we'll do is we'll add a salt. In this case, we frequently, for salting out, will use a salt of ammonium sulfate. So the reason salting out pulls proteins out of solution and makes some of them precipitate is essentially basically from what we talked about when we talked about the properties of water and solubility. So when you have a lot of salt in a solution, they'll sort of gather all these water molecules around them in a sort of shell that helps solvate them. And the more salt you add in, the more of the water component is sort of bound up and in dealing with surrounding all of the salt ions, and that makes less and less of it available to surround your protein of interest. And so, or any protein, not just your protein of interest. And so the protein, the less sort of naturally soluble your protein is, once you start adding salt, those proteins that have the least amount of solubility will start to precipitate out. So that's what's shown in this first step. You add a low concentration of ammonium sulfate, and then you'll get this precipitate. And this precipitate contains proteins, ideally, that are less soluble than your protein of interest. And then what you can do from there is centrifuge your tube, and then you'll end up with a nice little pellet at the bottom that contains a precipitate of proteins that you don't want. Your supernatant now contains your protein of interest, as well as other proteins that are just as soluble as your protein or more soluble. So at that point, you pull out just the supernatant, you put it in another tube, and then you add even more ammonium sulfate. So at this point, you have this really high concentration of ammonium sulfate. And now at that point, the precipitate that forms here is going to contain your protein of interest, as well as other similarly soluble proteins. What's going to remain in the supernatant are proteins that are even more soluble than your protein of interest. And again, you'll centrifuge it out, you'll get a pellet and a supernatant, and now this time your pellet is going to contain your protein of interest. So now at that point, we want to take the pellet, dissolve it in some fresh buffer, and then the next frequent common step that we'll use is dialysis. And dialysis, as we know, helps to allows water and salts and other really small molecules to move across the membrane. And so here we'll use dialysis to remove the ammonium sulfate from our target protein. And this is just a really common way to start generally purifying a protein away from a large mixture of proteins. So once we've completed dialysis, now we have a solution that contains our protein of interest, as well as probably some other proteins that have similar solubility properties. So at this point, we want to start using another purification method that will use maybe other properties to try to separate out our protein of interest from those other similarly soluble ones. And so a common purification step that will be used next is column chromatography. So in general, column chromatography, any column has two components. There's a stationary phase. The stationary phase is what sits inside your physical column, and it interacts with the proteins and everything else that flows through. And then you have your mobile phase. So your mobile phase is going to be that supernatant, right, that we got after our dialysis step. So it's got buffer, and it's got a bunch of proteins in it, including your protein of interest, but also maybe some other proteins that you don't want. And when you allow the mobile phase to pass through the stationary phase, that stationary phase ideally will help to separate out those different protein components that are in your mobile phase. So what are some of the different forces that we can use for column chromatography? So this is a list that goes sort of from low to high pressure, for lack of a better word. So the sort of simplest force you can use is gravity. You can allow gravity to allow your mobile phase to pass through the stationary phase of your column, and it can separate out your protein. It's a fairly slow process, and you'll often get large separations in between your proteins. So it's really great for sort of preparative methods where you want to physically separate out your proteins. And then going in order, we have these other methods. So next we have fast protein liquid chromatography, which uses a pump at sort of low pressure to push the mobile phase through the stationary phase. After that, we have high performance or high pressure liquid chromatography, HPLC, that uses even higher pressures. And then finally, we have ultra performance liquid chromatography or ultra high pressure liquid chromatography, UPLC or UHPLC. And so as we go up in force, we're able to do maybe better, more interesting analysis on our components of our protein, but it might be easier to separate out and get sort of preparative amounts of protein with using lower forces like gravity or the low pressure version of liquid chromatography. So that being said, let's talk about some different types of stationary phases that we can use. So one really common stationary phase or type of column chromatography that we use is called ion exchange chromatography, and this allows us to separate proteins by charge. So first we started out, just to summarize, we salted out based on solubility, and now we're going to start separating proteins by charge. So this is a basic setup for ion exchange chromatography. So we have our stationary phase, that's written here as our ion exchanger, that's the chromatography column, and it's going to bind to it's going to bind to proteins based on their charge. And then we add in our sample mixture, that's part of our mobile phase, and then we'll pass through an elution buffer, and we start with a low salt elution buffer. So, and then we'll start collecting fractions in these tubes. So we'll collect a little bit of liquid in each tube, and then we can make a chromatograph that essentially shows at what time and how much protein comes out in each of our tubes or fractions that we collect. And we can often measure this by UV absorbance, but there are also other methods you can use. So once we start passing through our low salt elution buffer, as our sample mixture passes down through this column, there will be different protein components that separate out. So we have this red protein. This red protein passed right through our column. It didn't interact with it based on its charge. It just started coming right through. And so in our chromatograph, we'll see when we added our low salt buffer, we see that our red protein came out. Then over time, we start adding a higher salt elution buffer. And the higher salt elution buffer will start pushing through some of these other proteins that were more strongly interacting with our ion exchanger column. And so the next one that's going to come through is our blue protein. That gets basically pushed out by the high salt in a sort of similarly actually to how salting out worked, where the salt sort of interrupts the interaction of the protein with the chromatography column itself. So we'll see our blue protein comes out, and then finally, as we keep pushing through more and more of this high salt buffer, last our yellow protein will come out. And so then we can see when we make this chromatograph, based on the different fractions that we collect, when these proteins elute out and how much of each protein we have. and that's based on their charge and their interaction with this charged column here. So there are a couple of different main types of ion exchange columns. As you might guess, there's positively and negatively charged ones, right? So first we'll discuss this anion exchange column, and that's named for the type of proteins it interacts with. So the actual column stationary phase is positively charged. And so it's going to interact really well with proteins that are negatively charged or anionic. So in that case, as you push through your low salt elution buffer, the beginning steps, the positively charged proteins, they'll come right through, right? Because they're not interested in sticking to our positively charged stationary phase of the column. And then over time, as we use our higher salt buffer, it'll sort of push those negatively charged proteins off the column and then finally allow those to elute out as well. And so the sort of opposite of that is our cation exchange column, where everything is flipped, right? So our stationary phase is negatively charged, and the column is named because it interacts with positively charged proteins or cationic proteins. Those cationic proteins will want to stick to the column and allowing the negatively charged proteins to elute through first in the lower salt buffer, and then as you add more and more high salt buffer, you'll sort of push your positively charged proteins off of the column and into the fractions that you collect. And something that's really important that can affect whether or not your protein is positively or negatively charged is, of course, the pH of the buffer that you put your protein in, right? So if you have a pH that's below the pKa of your whole protein in general, or the pi of your whole protein in general, you will have a protein that is positively charged. If you have a pH that's above the pi of your protein, you might have a protein that is negatively charged. But yeah, it depends on the individual properties of your protein, and the pKs, the individual amino acid makeup, and the pKs of each of those, and the overall pi of your protein. So so let's move on to our next type of column. So we just talked about ion exchange columns, which separate protons based on charge. Now we're going to talk about gel filtration and chromatography, where we separate proteins by size. So gel filtration chromatography uses this special type of basically little tiny beads or resin, and these beads are made of gel, and they have this gel matrix inside, and there's essentially little teeny holes or passages through the beads where smaller molecules, like very tiny proteins, can sort of get stuck inside these beads. They have to travel this little maze, so the pathway that the small molecules have to take is very long because they can sort of enter the gel, they have to travel around all through the gel, they get stuck, they have to turn around the corner before they can exit. And so to pass through a whole column full of these gel beads can take a really long time for the small molecules that fit inside. Whereas large molecules are large enough that they don't even fit inside these little gel passageways. They just flow right around the beads. So gel filtration chromatography is going to allow large molecules, large proteins, to come through first, whereas smaller molecules, smaller proteins will take a lot longer to be able to pass through. So here's an illustration of that setup. So we have our gel beads, that's our stationary phase, and then we have our solvent, our mobile phase, that's our buffer that's going to sort of go through with our mixture of proteins that we see over here. So, and then we, just like before, we collect fractions and we make a chromatograph. So as we push our solvent through, or as our solvent gets washed through the column, we can see our larger proteins, they're able to flow right through much more quickly, whereas our small proteins get kind of trapped near the top. And so over time, all of the proteins do eventually pass through the column, but first our larger proteins are going to elute out, which you would see first here in the blue. And then a while after that, finally, our smaller proteins can eventually make their way out. So you'd see later on, the later fractions, you'd see the red protein that's smaller. So that's a really great way to separate proteins by size. And there are different types of gel filtration resin that are most effective at separating certain sizes of proteins. So when you purchase gel filtration resin, you'll often see a number on it that'll indicate sort of what size proteins it's really good at separating from each other, depending on the size of the beads and the size of the gel matrix that's in the beads. So that brings us to our third type of chromatography, which is affinity chromatography. And this helps us separate proteins by their binding affinity to a binding partner that's on the stationary phase of the column. So a lot of times when we have our protein of interest, if we're separating it from a natural source, if we're just isolating it in its natural form, sometimes it will have some kind of natural component to it that's a really good binding partner for the type of affinity column chromatography stationary phase that's available. But a lot of times this is where our genetically engineered proteins come in. So what we can do when we're adding in the code for our gene of interest into a plasmid is we can also add on different types of tags that will help us down the road when we want to purify our proteins by making them by sticking on a really good binding partner onto the code for a protein of interest. So when our protein is transcribed and translated, it'll transcribe what's called a tag right next to our protein of interest, and then the whole thing will get translated as one protein that has your protein stuck covalently through peptide bonds onto a tag. And one really commonly used tag that we have here, that are shown here, is a his tag, which is a polyhistidine tag, and it has 6 histidine amino acids in a row. And so these histidine amino acids, let me just back up for a second, has the imidazole rings, right, as part of the side chain. They're full of all these nitrogens, and all these nitrogens, as we know, have all these lone pairs. And these lone pairs really love interacting with metals. So what we can use through affinity chromatography is a nickel column. So this illustrates here, this blue part is sort of the resin or the bead, but that is attached really strongly to a nickel ion. And so the nickel ion, when you pass the mobile phase containing your protein of interest through this type of column, the histidine tag will stick really strongly to the nickel. That's part of the stationary phase of the column. And all of your other proteins that have a much lower affinity for the nickel should pass right through the column. And so this is a really, really effective way of purifying a protein. A lot of times when you use those other types of column chromatography, you might have to use more than one column or more than one type of column to purify out just your protein of interest. But if you have a really successful affinity chromatography, you might only need to just do this one type of column to effectively purify your protein. But depending on the properties of your protein, how well it binds, and a number of other features like the folding, you may still have to do further purification steps after you do affinity chromatography. So again, here's an illustration of sort of how this works. So we have our column here. Our column has this solid resin matrix, and it has which is, this blue part that we have illustrated here. And then attached to it, we have our matrix anchored ligand, and that would be our nickel ion that's bound to this covalently, to this part of our resin. And then we have our mixture of proteins, and different proteins are going to have maybe different ligand binding site. Our protein of interest is the right shape to bind onto the matrix anchored ligand. So you can see as you pass this mixture of proteins through the column, your protein of interest that has that binding site, in our case the histidine tag, is going to stick to the column. All the other proteins are going to pass through because they're not interested in sticking onto the nickel. Then finally, once you've discarded all these other proteins, you are ready to elute out your protein of interest. And so basically to do that, you have to unstick, in this case, you had to unstick the histidine tag from the nickel. And so often what people will do here is you'll add a high concentration of imidazole, and the imidazole itself will stick really strongly to the nickel and sort of dislodge the histidine tag with your protein and allow your protein to flow through. Another thing you could also do potentially is to add in a bunch of nickel ions that will bind to the histidine tags of your protein, essentially doing the same thing, kicking it off of the resin and allowing the protein to flow through so you can collect it. So that being said, now we've learned a number of different ways to purify proteins based on solubility, size, charge, affinity to binding partners. So you're ready to go ahead and answer those protein purification practice questions.